Meiotic cell division takes place in the gamete-producing cells in certain organisms, such as humans, so in the testes and the ovaries. It's used for making sperm and ova. During interphase, the chromosomes, they have to make an extra copy of themselves, just like that in mitosis. So there is DNA replication during interphase. The cell will then undergo two divisions, meiosis 1 and meiosis 2. Now let's look at meiosis 1. During the prophase of meiosis 1, you will see that the homologous chromosomes, they pair up together. This does not happen in mitosis, but in meiosis it does. By the way, the blue one is from the father and the red one is from the mother. Then in metaphase 1, the homologous chromosomes, they will pair up and line up at the same time. So they would line up in pairs in double file in the middle of the cell. During anaphase, the homologous chromosomes will separate. This is different from in mitosis. In the case of mitosis, it is the sister chromatids separating. But in the case of meiosis, it is the homologous chromosomes separating. The effect of this on the cell is that during telophase 1 in meiosis, you will have one member of each homologous pair in each cell. This is very different than in the telophase during mitosis. Now let's compare the two. During mitosis, the chromatids have already separated, but in telophase 1 in meiosis, it has not. Also, at the end of mitosis, the daughter cells are identical to the parents. But in telophase 1 in meiosis, there is different genetic makeup in the daughter cells. Now the process in meiosis 2 will be quite similar to that in mitosis. This is prophase 2, so you will see that the nuclear membrane is integrating. At metaphase 2, the chromosomes will line up at the equator. At anaphase 2, it will be the sister chromatids that are separating, so similar to mitosis. At telophase, what you will see is that the new nucleus have already been formed. There will be different genetic makeup in the daughter cells. Also, you will see that the number of chromosomes in the daughter cells are halved of the original. If you still remember, in the original cell in the testis or in the ovary, there are 6 chromosomes, or 3 pairs. But now after meiosis 2, there are only 3 in each nucleus. So how does meiotic cell division cause genetic variation? The first way is by independent assortment. Now whether the blue one is on the left or is on the red is totally random. And how the first pair arranges itself is independent of how the second pair arranges itself. There can be many different combinations. This causes genetic variation. The second way is by crossing over. In prophase 1, when the homologous chromosomes pair up, they will stand next to each other and they may touch. When they touch, there may be an exchange of genetic material. When genetic material has been exchanged, then there will be new combinations on a single chromosome, and this can cause genetic variation.